presented by Cameron Lee, and the title of the talk is Exploration of Gene Expression in the Burmese Python, or Python vivittatus.
Well, I feel like I should say that I hope the dog's okay. So, like she said, my name is Cameron Lee, and we're not a computer programming class. We actually study gene expression in the Burmese python. It's a really incredible animal, and what I find fascinating about it is this is not actually as big as it can get. This one is 15 feet, and it was found in Florida, but they can get up to 23 feet and 200 pounds, which I find pretty terrifying personally. So before we dive into the research, I'd like to give a little background on the Burmese python, as well as its relation to human disease, and then in part 2 of the presentation, I'll talk about the experimental methodologies that we use, as well as the results and conclusions. So the Burmese python, as you could probably imagine, got its name from Burma, which is now called Myanmar. Even though it's named after a small country, the breadth of its native habitat stretches from northern India all the way down to the middle of Indonesia. The brown points here represent the population distribution of the Burmese python, which is the python of our focus. So in the last couple decades, the Burmese python has established itself as an invasive species in the United States. It was first introduced into Florida during Hurricane Andrew in 1992, and what happened was the hurricane destroyed exotic pet stores as well as zoos, allowing it to escape and get into the wild. The Burmese python can live in most places, and as you can see here, the green represents suitable habitat that it could live, so the U.S. Wildlife and Fisheries Service made it illegal to transport them across state lines. What's shocking is that it covers almost the entirety of every state on the southern U.S. border. So a lot of people ask, why a python? How could a python ever actually contribute to modern medicine? Well, the Python Project got its start with the understanding that the Burmese python is an extreme model of physiology. The postprandial Burmese python boasts metabolically active organs that undergo drastic growth and regression with respect to digestive periods. The heart grows by a factor of about 1.58, and the liver almost doubles in size. What's important to note about this is that the growth is physiological. It's actually healthy for the python, and it helps it meet the metabolic demand of the new meal that it just ingested. So on the top here, you can see that incredibly the python is able to ingest a meal whole and then completely digest it in a 6-day period. Below that, you can see the lipid serum concentrations throughout the digestive period, with 1-day post-fed being the highest for it. What's important to note here is these concentrations in the python would actually be lipotoxic and lethal in people. So determining how the python is able to remove these lipids could someday lead to disease therapeutic development. So there's 3 types of disease that I want to talk about today. Both of them are all 3 of them are either caused by indirectly or directly consistently high concentrations of lipid in the blood serum. The first of which is non-fatty or non-alcoholic fatty liver disease, which affects approximately 20% of the United States. It's characterized by the deposition of excess lipids into the liver. Okay. So, as it goes on, liver cirrhosis can develop where healthy hepatocytes are actually replaced by scar tissue. As you can imagine, scar tissue has trouble meeting the physiological demands of healthy parenchymal cells. Liver cirrhosis kills approximately 27,000 people every year, and I feel that it's important to reiterate that the python's lipid concentrations would actually be lethal in people. So another big hitter is coronary artery disease. It's brought on by the buildup of fatty acids and calcified plaques in the arteries. It's one of the biggest killers in modern society today, killing roughly 7,000 people every year. With the diseases being brought on by consistently excessively high lipid concentrations in the blood, you can see why it might be important to determine how the python is able to remove such large amounts of lipid. So to address this question, the Python Project decided to study the gene expression of genes that are critical to or associated with lipid metabolism. So to start, the Python genome is largely unannotated, and we don't know much about it. So we had to design gene-specific primers through a homolog in an evolutionarily related species. From there, we created a standard curve using qPCR, and then as things started to pick up, RNA was isolated from python liver tissue at fasted and varying intervals post-fed. After that, cDNA was synthesized, and I'm just going to keep going. Quantitative PCR was used to amplify and quantify the cDNA in real time. So the aim of our research is to determine how the python is able to remove such high concentrations of lipid. All excess cholesterol in your body is processed in the liver and is converted to bile. The pathway for the bile acid synthesis is extremely complicated and is regulated by over 100 genes. The genes regulate the moving of lipids into the liver, the processing of them into bile, and then their removal from the liver. So So there's a graph here. Oh, there we go. Okay. So over the past 3 years, the Python project has studied 96 of these genes, and we've successfully found expression in 66 of them. In the graph that is supposed to be right here, you see that lipid transport and bile acid synthesis are at their highest expression levels at 1 day post-fed. At 3 days post-fed, you see bile transport at its highest expression level. The graph on the right here represents averages of the gene expression. So lipid transport in orange and bile acid synthesis in gray are at their highest expression levels at 1 day post-fed. And like I said, bile transport in the rust color here is at its highest level in 3 days post-fed. Now what's really incredible about this data is that it actually agrees with the change in concentrations of lipids during the 6-day digestive period. So you can see here when lipid concentration is the highest in the 1 day post-fed, the things that you would expect to be expressed the highest, like lipid transport and bile acid synthesis, are expressed expressed at their highest point to accommodate the high levels of lipids. Bile transport then, at 3 days post-fed, corresponds when much of those lipids have been converted to bile, and now it needs to transport them out of the liver. So it makes sense that the bile transport would be at its highest expression. So with results as exciting as these, it's important to think about where the Python project should go in the future. There's a lot of options. One of them could be the detailed analysis of the relationship among these genes. Another could be the examination of hepatic tissue from overfed pythons. A third could be measuring candidate genes in diseased and healthy human liver. So before we get any questions, I would like to make some acknowledgments. I'd like to thank Leslie Lineman, Angela Peter, members of the Lyman Lab, the MCDB department, and all of those whose contributions and hard work have made the Python project possible. And one last thing, Dr. Harvey sent this to me a couple days ago. She found this and it looks just like the normal digestion, but instead of a rat, it's actually an alligator, which to me is terrifying. So if anyone has any questions, I'd be happy to do my best to answer them. Yeah. It's a good question. I think it digested it first and then... Beyond that, I got nothing for you, so... Alright. Alright, well, it's... Oh, yeah.
So, most of the genes looked at were found using homologs, but it seems like the Burmese python is very unique in its ability to metabolize fats. Is there genes you didn't look at that could have to do with?
It certainly could be possible. The genes that, or the ones that we found in the python were from evolutionarily related species, so they're largely conserved. They could just be expressed more if I had to take a stab at it, but I'm sure there are other genes out there too. So. All right, well, hey, it's been great. Thank you.